which clustering method is most suitable for analyzing 16S data when a significant proportion of bacterial species in the samples are unknown? So the answer is de novo. Best clustering method when species in sample are unknown. So de novo clustering groups reads by similarity. Without reference sequences. When many taxa are novel, not in reference databases, de novo retains all reads and clusters them together, capturing unknown diversity. In contrast, closed reference clustering maps reads only to closed reference clustering maps only to known references and discards unmatched reads. It would lose novel taxa entirely. Then open reference first maps known, then clusters the rest. It still relies partly on references. Thus, to include unknown lineages, we need de novo clustering. Now, option D, hybrid is not standard terminology. Open reference could be considered hybrid, but even that depends on reference first. So the answer, the most suitable one is de novo. Now, in metagenomic study, you have two samples, sample A and B. Sample A has a mean sequence depth of 50x, while sample B has a mean sequence depth of 200x. Which of the following statements is most likely true? Sample B contains a more diverse microbial community than sample A. This is true because it has greater mean sequence depth than A. Now, option B is the least strong of the choices. Higher sequencing depth in sample B means more reads and a greater chance to detect rare taxa. So, more diverse microbial community. In practice, deeper coverage can reveal more species, making observed diversity appear higher. Thus, B contains a more diverse community could be interpreted as B shows higher observed diversity due to deeper sampling. Right? Now, answer A, let us check for that. Sample A is of higher quality than B. So, answer like option A is wrong because depth is not a proxy for data quality. Both could be high quality regardless of depth. And C, sample A is more suitable for detecting rare microbial species. That is also wrong because both could be high quality regardless of depth. So, sample B, not the shallow A, would be more suitable for rare taxa. Now D, sample B has a lower total number of sequencing reads compared to sample A. So if B has 200x and A 50x, B actually has more reads, contradicting D's claim. In fact, the info content, that is the number of reads, is usually proportional to depth, right, for the similar samples. Note, real diversity is a property of the community, not sequencing effort. None of the statement is strictly universally true. However, by convention, deeper sequencing will capture more species, hence more diversity. That is why B is the answer. Now, we're moving on to section B, single word or very short answers. Why barcode is attached to sequence before sequencing? The answer given is to enable the identification and sorting of different samples or molecules within a single sequencing run, facilitating multiplexing. Barcode is attached to a sequence before sequencing to enable identification and sorting of different samples or molecules within a single sequencing run, facilitating multiplexing. Right. So barcodes or indices are short DNA tags attached or added to each DNA library before pooling. They allow multiple samples to be sequenced together, multiplexing in one run. After sequencing, the barcode sequence is read and used to demultiplex the reads, assigning each read to its original sample. In practice, indexing greatly increases throughput and cuts the cost. Many libraries are pooled, sequenced, and then sorted by their unique barcodes. Each reads barcode labels it so that bioinformatics pipelines can split pool data back into per sample FASTQ files. Now, barcoding enables identification sorting of reads by sample, facilitating cost-effective multiplex sequencing. Now, Bellerophon is based on which approach from the algorithmic viewpoint to detect chimeric sequences? So the answer is tree-based. Now, let us discuss this in depth. Bellerophon chimera detection is based on what approach? Bellerophon uses a phylogenetic or partial tree approach. It splits each candidate sequence into two overlapping fragments, builds distance matrices or trees for each fragment, and looks for phylogenetic incongruence between the halves. If the left and right halves of a sequence cluster differently, as if from different parents, it's flagged as a chimera. In short, Bellerophon's algorithm compares tree topologies or distance matrices of sequence fragments to detect chimeras. This partial treeing method comparing subtrees is why we call it tree-based. Other chimera tools use abundance or reference comparisons, but Bellerophon explicitly infers phylogenetic relationships within sequence fragments. The process of separating mixed DNA sequencing reads into individual samples based on their unique barcode or index sequences. Process of separating mixed DNA sequencing reads into individual samples based on their unique barcode or index sequences is known as demultiplexing. So, process of separating pooled reads by barcode into individual samples is demultiplexing. After sequencing, the pipeline reads each fragment's barcode and sorts reads into separate files for each sample. In other words, one takes the mixed data from all the pooled libraries, and splits it back into per sample FASTQ files based on barcode sequences. So, demultiplexing would ensure that each read is correctly assigned to its source sample or library. On Illumina runs, this often happens automatically on instrument. On other systems or custom barcoding, for example 10x Genomics, software scripts match barcodes to libraries. Either way, demultiplexing segregates reads by their barcode identifiers. Now, what is compositional data? Compositional data is data where all values sum to a constant. data where all values sum to a constant. So it is basically compositional data are proportions or counts that carry only relative information and are constrained by a constant sum. For example, a microbiome samples OTU or ASV table is compositional because counts are often normalized so that the total per sample is the same. For example, 100% of fixed sequencing depth. Thus, the components taxa are summed to a constant, often 1 or 100%. Now, in compositional data, only relative differences matter. One taxon's abundance is meaningful only relative to others, and standard Euclidean stats can be misleading. This is why special transformations, CLR, Hsson geometry are used. In summary, compositional data implies values whose sum is fixed, encoding relative rather than absolute amounts. Euclidean distance to CLR transform values is called Hsson distance. So Euclidean distance computed on centered log ratio transform compositional data is known as Hsson distance. In compositional data analysis, this metric appropriately measures differences between samples because CLR removes the constant sum constraint. Mathematically, Hsson distance is Euclidean distance on the CLR vectors named after Hsson's work on compositional data. Now, next question is dash refers to the union of all genomes of a given species or a taxonomic class or taxonomic clade. Union of all genomes of a given species or a taxonomic clade is pangenome. So pangenome basically, or a supragenome of a clade, is the set of all genes present across all strains of that clade. It includes the cogenes that is found in every strain plus the accessory genes present in some strains. In other words, the pangenome is the complete genetic repertoire of a species or a clade as the union of individual genomes. Right? Now, dash and dash distances are based on differences in the ranks of different taxa within samples. The answer is Kendall and Spearman. So these are Kendall's tau and Spearman's rho or Spearman's rank distances. Both are rank-based correlation metrics. Spearman's distance relates to the Pearson correlation of rank counts, and Kendall's tau counts the number of pairwise rank inversions between samples. In ecology, they measure how similarly two samples rank taxa abundances. Both ignore absolute values and depend only on the ordering of taxa. Now moving on, similarity matrix based on the intersection over union of taxa is known as Jacquard similarity. So basically, let me write the formula as well for you. J is equal to the Jacquard formula, 1 minus A intersection B, that is the intersection of these two sets, that is the species that are common to both, and then we basically put the mod, that is because we are taking the number and this is a numeric value, right? So A intersection B, basically the number of species that are common to A and B, that is this area, and the union, that is the combined of A and B, right? Both the number of species in A as well as B, A or B. This is A or B and this is A and B, and we subtracted with 1. This is Jacquard value, and that's how we calculate it. So for two samples, Jacquard similarity is equal to the size of intersection of taxa lists upon the size of union of taxa lists, and we need to subtract 1, right? It ranges from 0, no taxa in common, to 1, that is these are identical taxa sets. In microbiome context, a Jacquard similarity matrix shows pairwise similarity based on shared taxa presence or absence. Now our second last question is DARA2 is a popular tool for classification of ASVs. The tool is based on the modified Needleman-Wunsch algorithm. Needleman-Wunsch algorithm, basically NW global alignment algorithm. In fact, during sample inference, DARA2 uses a banded NW alignment to compare each candidate sequence against error models, counting mismatches and indels. This alignment step is Needleman for identifying true sequence variance from errors. Thus, the answer is Needleman-Wunsch. Now, finally, which line contains the Fred quality scores in a FASTQ file? That is the fourth line. Fred quality scores Q is equal to the formula, right, that we just discussed. Minus 10 log base 10 and the probability, right? Minus 10 log base 10 probability of error. This is the formula. Now FASTQ entry containing the Fred scores is fourth line. Each read is of is 4 lines. The line 1 is the header starting with at the rate. Line 2 is the nucleotide sequence, and then line 3 is the separator plus, and line 4 contains the quality scores encoded as ASCII characters. Thus the Fred or the quality scores are on fourth line of each FASTQ record. So that's it.